Let's start nucleic acid sequence database EMBL. Nucleic acid sequence database is a database which contain the information regarding the nucleotide sequences DNA and RNA. There are total three nucleotide sequence databases: GeneBank at NCBI, EMBL at EBIN, DDBJ Japan. All the three organizations together form INSD National Nucleotide Sequence Database Collaboration. They coordinate among themselves and exchange data on daily basis. This means if you submit your sequence to EMBL, the EMBL will exchange that data with GeneBank and DDBJ and vice versa. In this way, all the three organizations are updated with the latest finding. Today we will start with Nucleic Acid Sequence Database EMBL, that is European Molecular Biology Laboratory, is a primary nucleotide sequence resources maintained by European Bioinformatics Institute situated in United Kingdom. It is a molecular biology research institute supported by 27 member states. EMBL was created in 1974 and has become Europe's flagship laboratory for the basic research in molecular biology. DNA and RNA sequences are submitted directly from the individual researchers, genome sequencing project, and patent application. EMBL groups and laboratories perform basic research in molecular biology and molecular medicines, as well as organizes training for scientists, students, and visitors. The database is a part of international collaboration with DDBJ and GeneBank. Data is exchanged between the collaborating database on daily basis. In this diagram, I have shown you how the data is exchanged between these three organizations. Okay, these three together form INSDC, that is International Nucleotide Sequence Database Collaboration. The web-based tool Webin is preferred system for individual submission of nucleotide sequence, including third-party annotation and alignment data. Webin is the tool for the submission of nucleotide sequence to EMBL. In EBI, Sequence Retrieval System (SRS) integrates and link the main nucleotide and protein database as well as many other specialist molecular biology databases. SRS is the search engine of EMBL EBI. SRS is a type of integrated search engine. Integrated search engine means it integrates all the databases of EMBL. Okay, in EMBL, I think there are 29 databases, means all the 29 databases are interconnected. Sequence similarity searching, a variety of tools, for example, FASTA and BLAST are available that allow the external user to compare their own sequence against the data in EMBL and other databases. FASTA and BLAST are the sequence similarity tool. What the FASTA and BLAST will do, it will compare your query sequence with all the sequence present in the database, and it will give you the top 100 sequence which is similar to your query sequence. Okay, all the available resources can be accessed by EMBL homepage. The flat file entry contains the sequence and the brief description for cataloging purpose, the taxonomic description of the source organism, bibliographic information, and the feature table containing the location of coding region and other biologically significant site. हर एंट्री का क्या रहता है, दो सेक्शन रहते हैं. वन इज द सीक्वेंस सेक्शन विच कंटेन्स द न्यूक्लियोटाइड सीक्वेंस एंड अनदर इज द एनोटेशन सेक्शन. Annotation section will contain the taxonomic description of the source organism, bibliographic information, and the feature table. Feature tables में क्या रहता है? The feature table will contain the information derived from the sequences such as the coding region, exon, intron, gene name. ये सारी चीजें जो है आपकी feature table में आती है. So the typical entry will contain the sequence section and the annotation section. So this is the content and the format of entry. It is which starts with the word ID and end with double slash. Okay, the ID stands for identification. Inside the ID there are 6 subfields. One is the identification name, which is similar to that of the accession number. Next is the topology, linear or circular. है ना, यहाँ पे जैसे लीनियर है आपका जो भी सीक्वेंस जिसका भी है आपका डीएनए का सीक्वेंस है, the linear, then the molecular type, genomic DNA, then the data class, then data division, and then the base count. These 6 subfields together form identification. AC stands for the accession number. Accession number is a unique identification number given to the sequence entry in the database. Then DT is the date at which the sequence was created and at which the sequence was submitted to the database. Second DT is the date at which the sequence was last updated. DE stands for description. KW stands for the keyword, stands for the organism source. OC stands for organism classification. Then RN is the reference number. Okay? A single entry may contain multiple references. So this is the reference number 1, then reference number 2. Then this is the reference page number. RA stands for the author's name, then the title, then the journal's name. Then DR is the cross references. FH is the feature table. Okay, FT is the feature table. Exon, intron, CDS, what all you know about your sequence, you can give in the feature table. Then this is the sequence which starts with the word SQ and end with double slash. Okay, this is the format entry of EMBL, which starts with the word ID and end with double slash. Here I have shown you the description of the flat file information. ID stands for identification, AC for accession number, DT for date, DE for description, GN for gene name, OS for organism species, originally, organism classification, reference number, reference position, reference comment, cross references, RA for reference author, RL for location, CC for comment, DR for database cross references, KW for keyword, FT for feature table, SQ for sequence header, and double line for termination line. Okay, some entry do not contain all the line type, and some line type may occur many times in the single entry. It is not necessary that all the line type must be present in the सिंगल एंट्री है ना, जैसे ये एंट्री जरूरी नहीं है कि इसमें वो सारे के सारे लाइंस हो, एंड सम लाइंस आर रिपीटेड multiple times. For example, reference number, here the reference number are 2 times. Okay, feature tables are multiple times, DR for 3 times. So some lines may occur many times in the single entry, but each entry must begin with the identification line ID and end with the termination line double dash. Okay, हर entry जो है आपकी starting जो रहेगी वो ID से होगी and it will end with double dash. Then EMBL data classes shows the EMBL data classes to which the record belongs and is indicated by the 3-letter abbreviation. Okay, this is for stands for raw expressed sequence tag. WGS stand for genomic context, GSS for genome survey sequence, STD for the assembled and annotated sequence, then PAT for the patent sequence, EDS for the coding sequence. ठीक है, ये डेटा क्लास कहाँ पे रहता है आपका? देखो ये जो सीक्वेंस एंट्री का जो आईडी है उसी में आपका ये डेटा क्लास रहता है. ठीक है, ऐसे. And this is the home page of EMBL EBI. Okay, this is the home page, okay, which contain the search box where you can text your query. Then from here you can select the databases. Then in the services you can find the data resources and tools of EMBL EBI. For that you have to click on services. To see the ongoing research on EMBL EBI, you can click on research. To see the training programs of EMBL EBI, you can click on training. And to study about EMBL EBI, you can click on about us. The same thing is present here also. To find data resources, to submit data, you have to click here. To explore the research on EMBL EBI, you can click here. And to see the training program, you can click on train with us. Okay. So this is the home page. Here you have to type your query. Here I have written the accession number. Then from here you can select the database from where you want to extract the information. अगर all रखोगे तो ये सभी में search करेगा, or you can give any specific database also. Okay. On clicking on search, it will give this results. Okay. The information regarding Z92910 is present in this 5 databases genome and metagenome, nucleotide sequence, protein sequence, gene expression, and EMBL EBI website. Okay, from here you can filter out your result. अगर आपको genome में जाना है तो आप इसको select करोगे. If you want to see the nucleotide sequence, you can click on the nucleotide sequence. If you want to see the protein sequence, you can click here. If you want to go to the gene expression database, you can click here. Okay. Then here I have selected the nucleotide sequence. Okay, in the nucleotide sequence, after selecting the nucleotide sequence, you can see even though you are on the nucleotide sequence page, you will find the cross references link for the different databases like gene expression, literature, genome, and metagenome. This means that the search engine of EBI is an integrated search engine. Okay, even though you are on the nucleotide sequence database, it is showing you the cross references link. Okay, you can extract the sequence in either in the EMBL format or in the FASTA format. Okay, then after selecting this Z92910, it will open up this page. ENA, that is a European nucleotide hierarchy. This is the nucleotide databases of EMBL EBI. Okay, this is the description of the sequence Z92910. The name of the organism from where this sequence have been extracted is Homo sapiens. The accession number is Z92910. The molecular type is the genomic DNA. The topology is linear. Base count is 12146. Data class is STD. The data division is HUM. The keyword is hemochromatosis HFEG. The chromosome number is this, and this is the software. Now you can view the EMBL or the FASTA format. You can download the sequence format. You can navigate the page. Means if you click on this show button, it will show you the cross references. Okay. Then you can go to the publication and on which this particular sequence have been published, and you can see the sequence version. On clicking on sequence version, it will open up this page, the sequence version R key. Okay. The accession number, accession version, sequence version, first published date, last updated. Then you can view the sequence in EMBL or in the FASTA format. You can also download the sequence. On clicking on EMBL, it will open up this EMBL format, which starts with the word ID and end with double dash. Okay, this is the annotation section, which gives the feature of the sequence. And this is a sequence section which starts with SQ and end with double dash. Okay. If you click on FASTA format में अगर आप click करोगे तो the FASTA format starts with a sign greater than. Then there is a small description about the sequence. यहाँ पे अपन ने ENA, accession number, accession version and the source. Then there is a sequence. This is the FASTA format. Okay. Then if you click on navigation, it will show you the cross references links. Okay, means the information of Z92910 is also present in taxonomic database, assembled GN, assembled TR, Europe TMC, which is a literatural database, and in IMGT and HLA database. Then if you click on the publication, it will show you the article on which this particular sequence has been published. You can see the abstract. You can go to the DOI. Okay, this is how to search on EMBL EBI. Then the sequence submission in EMBL, Webin is the tool by which you can submit your sequence to EMBL. Webin is a submission tool for the nucleotide sequence and their biological annotation information to EMBL bank. The Webin is designed to allow the fast submission of the single or the multiple number of nucleotide sequences. All the information required to create a database access could be accumulated during this submission process, such as submitter data, launch date information, sequence statistics, description, source information, reference quotation information, etc. Now, all the information which is required to create the database access are accumulated during the submission process. And when you submit your sequence to the Webin, it will give you the steps during which you will submit all the information which is required to create the database, like submitter data, जिसमें आप contacts डालोगे अपने, then launch date information when you want to publish sequence. Okay, then sequence statistics, description, source information, reference information, all this information are accumulated during the submission process only. Webin is available from the EMBL www homepage or at URL this. The sequence annotation in Webin is added from the summary and sequence feature page. Any number of relevant feature can be easily added to the sequence feature table from the comprehensive list and by filling out the specific feature form. To add annotation, you have to go to the summary and sequence feature page. Okay, from where you can add any number of feature to your sequence. To assist the submitter in selecting the feature for the sequence, the web feed provide a full description of all the EMBL features and qualifiers, where the EMBL annotation example illustrate how these features and qualifiers should be used within the standard EMBL entries. Okay, if you don't know how to select the feature or the qualifier for your sequence, you can go to the web feed. Okay, the web feed will provide you the full description of all the EMBL feature and qualifier. वहाँ पे आपको सारे फीचर्स क्वालिफायर्स उसका डिस्क्रिप्शन मिल जाएगा विथ एग्जांपल which will help you to select the feature and qualifier for your sequence. Okay, if you want to submit your sequence to ENA, then go to the submit to ENA. This will open up this page, submitting and updating data. Okay, this is you can submit to ENA either by using the Webin portal or by using the Webin CLI. Webin portal is the online submission, web-based submission, and Webin CLI is a standalone submission where you have to make the document in EMBL format, and then you have to email it to EMBL. Okay, the CLI will help you to create the submission file. ENA allow the submission by the three routes, each of which is appropriate for different set of submission type. Okay, first is the interactive submission, second is the command line submission, and third one is the programmatic submission. The interactive submission are completed by filling out the web page directly in your browser and downloading template spreadsheet that can be completed offline and uploaded to ENA. This is often the most accessible submission route. Okay, the interactive submission is a Webin portal. You can use Webin portal for it. It is a web-based. Directly, you can submit your sequence to EMBL by using your browser, and you can download the template spreadsheet. Template spreadsheet is for the feature table, yeah, which you can complete offline and then you can upload it to ENA. The second is the command line submission, which uses a bespoke Webin CLI program. This validates your submission entirely before you complete them, allowing you maximum control of the process. The command line submission will help you to create the submission file, which you have to submit it to ENA. The third one is a programmatic submission, are completely completed by preparing your submission as XML document and either sending them to ENA by using programs such as URL or by using the Webin portal. Now, programmatic submission, you have to create your submission file as a XML document and then you have to send it to ENA by using URL or by using the Webin portal. This is the table which outlines what can be submitted through each submission route. For the study data, you can go for the interactive submission or to the programmatic submission. You can submit the sample data either by interactive submission or by programmatic submission. The read data can be submitted by all the three routes. The genome assembly data can be submitted by Webin CLI. The transcriptomic assembly data can be submitted by Webin CLI. The template sequence can be submitted by Webin CLI, and the other analysis data can be submitted by programmatic route. So this is the table which outline what you can be submitted through each submission route. This is about DDB EMBL. Now I will show you the same thing in by using internet. Okay, for searching on EMBL using internet, you have to write EMBL EBI on Google and then click search. This will open up this page. Then select EMBL EBI home page. Okay, this will open up this page. This is the home page of EMBL EBI. Okay, to see the services, means the data resources and tools of EMBL EBI, you have to click on services. Okay, see, explore all our data resources and tools. Okay, the EMBL EBI maintains the world's most comprehensive range of freely available and up-to-date molecular data resources. You can sort by alphabetical A to Z or Z to A order. Then you can refine by टाइप. आपको डेटा रिसोर्सेज देखना है या फिर आपको टूल्स देखना है या फिर आपको दोनों देखना है, आपको सेलेक्ट कर सकते हो यहाँ पे. एंड हियर यू कैन सेलेक्ट द कैटेगरी. ठीक है, आपको डेटा रिसोर्सेज केमिकल बायोलॉजी से रिलेटेड देखना है, क्रॉस डोमेन, डीएनए, आरएनए, जीन एक्सप्रेशन, लिटरेचर ऑन system, you can filter. Like if I select to the literature, so there are three data resources in EMBL EBI regarding literature. एक है आपका यूरोप PMC, एक है आपका यूरोप PMC manuscript submission, and the third one is the patent database. Below each database, you will find the small description which will explain about that database. If you don't know about Europe PMC, by reading this, a database to search the worldwide life science literature. Right? It is a literatural database. Now, a manuscript submission is a self-deposit full text article. It will contain the full article. Okay? Patent database is a non-redundant patent sequence database providing access to full text patent document. It will contain the information regarding the patent document. Okay. Similarly, if I click on the protein, it will open up the data resources regarding the protein information. Like AlphaFold DB is a database for the protein structure prediction for the numerous species. UniProt is a comprehensive resources for the protein sequence and functional annotation. PFARM is a database of conserved protein families and domains. PFARM is a member database of Interpro. है ना? का हो गया. वैसे सेम अगर हम डीएनए आरएनए देखना चाहेंगे तो वी कैन क्लिक ऑन डीएनए एंड आरएनए. तो असेंबल जीनोम ईएनए, ये सारा आपका ओपन हो जाएगा. देन इफ यू वांट टू स्टडी द टूल्स, ठीक है, टूल्स क्या रहेगा, दे यूज फॉर द एनालिसिस ऑफ योर ओन डेटा. ठीक है, जैसे क्लस्टाल ओमेगा. क्लस्टाल ओमेगा क्या है आपका मल्टीपल सीक्वेंस अलाइनमेंट टूल है. इट इज अ मल्टीपल सीक्वेंस अलाइनमेंट ऑफ डीएनए एंड आरएनए प्रोटीन सीक्वेंसेस. क्लस्टाल ओमेगा रिप्लेसेस द ओल्डर क्लस्टाल डब्ल्यू अलाइनमेंट टूल. The multiple sequence alignment tool is used to find the conserved domain of the sequences. ना, इसमें हम क्या करते हैं, एक ही फैमिली से, जैसे आपने एक ही प्रोटीन फैमिलीज के आपने 10 सीक्वेंसेस लिए एंड यू वांट टू नो द कंजर्व्ड पार्ट. कंजर्व्ड पार्ट मतलब कि जो इवोल्यूशन के दौरान इनमें कोई भी चेंजेस नहीं आए, उनको हम बोलते हैं कंजर्व्ड पार्ट, है ना. तो हम उसको जब क्लस्ट्रल ओमेगा में आप जब डालोगे तो क्लस्ट्रल ओमेगा क्या करेगा, वो 10 के 10 सीक्वेंस को आपस में कंपेयर करता है, होमोलॉजी बेसिस पे वो कंपेयर करेगा एंड इट विल शो यू द पार्ट विच इज कंजर्व्ड, है ना, जो आपका चेंज नहीं है, जो सेम है, वो आपको वो पार्ट दिखाएगा. तो अब हम बोल सकते हैं कि दैट पार्ट इज द कंजर्व्ड पार्ट विच इज नॉट चेंज ड्यूरिंग इवोल्यूशन. तो ये आपका क्लस्ट्रल ओमेगा का फंक्शन है. दिस अ मल्टीपल सीक्वेंस अलाइनमेंट ऑफ डीएनए एंड प्रोटीन सीक्वेंस. ब्लास्ट इज अ फास्ट लोकल सिमिलैरिटी सर्च टूल फॉर द प्रोटीन सीक्वेंस डेटा, है ना. ब्लास्ट क्या करता है, आपका जो क्वेरी सीक्वेंस है, इट विल कंपेयर योर क्वेरी सीक्वेंस विद ऑल द सीक्वेंसेस प्रेजेंट इन द डेटाबेस एंड इट विल गिव यू द टॉप 100 सीक्वेंस व्हिच इज सिमिलर टू योर क्वेरी सीक्वेंस. ये क्या करता है, आपका क्वेरी सीक्वेंस को पूरे डेटाबेस के सीक्वेंस के साथ कंपेयर करता है ब्लास्ट. तो दिस ऑल आर द टूल्स present in the EMBL EBI which you can use for the analysis of your own data. Okay. Then next is the research. From here you can see the ongoing research on EMBL EBI. The European EMBL EBI has a global leader in a computational biology research since 1994. You can explore the research group of EMBL, EBI. These are the news, research news है ना, जो भी आपका publication है EMBL, EBI का, वो आपको यहाँ पे आप देख सकते हो. Research achievements, then research partnership of EMBL, EBI, ये सब चीज़ें हम यहाँ पे देख सकते हैं. Then you EMBL, EBI training, delivering a world-class training in the data-driven life sciences. Okay? They also uh, provide training to the students, scientists and visitors. तो अगर आपको ट्रेनिंग करना है EMBL, EBI में, तो you can click on training. These are the courses where you can register. ठीक है, लाइव वेबिनार, वर्चुअल कोर्स एट EMBL, EBI. यहाँ पे आपका एप्लीकेशन फॉर्म भी रहता है जो आप जिससे आप यहाँ पे रजिस्टर कर सकते हो. इफ यू वांट टू स्टडी अबाउट EMBL, EBI, देन यू हैव टू क्लिक ऑन अबाउट अस. ठीक है, एट EMBL, EBI we help scientists release a potential of big data and biology, exploiting the complex information to make discoveries that benefits humankind. Who we are, our missions, our funding, our funders, our story, बायोइन्फॉर्मेटिक्स टुडे, why बायोइन्फॉर्मेटिक्स मैटर्स, है ना, ये सारी चीज़ें आपको we can study about EMBL, EBI by clicking here. Okay, coming to the home page on EMBL, EBI, this is the search box where you can text your query. From here you can select the database from where you want to extract the information. अगर अपन ऑल रखेंगे तो वो सब में सर्च करेगा. अगर आप कुछ स्पेसिफाई कर दोगे तो वो उस स्पेसिफिक डेटाबेस में सर्च करेगा. आप यहाँ कैट बाफना भी डाल सकते हो. कैट इज द नेम ऑफ द जीन, बाफना इज द नेम ऑफ द ऑथर. और यू कैन सर्च बाय एक्सेशन नंबर. जैसे Z92910 को अगर मैं सर्च करती हूँ, ठीक है, तो see, the information regarding 92910 is found in this 4 databases in the genome, metagenome, nucleotide, protein and gene expression database. From here you can select your data. आप यहाँ पे select कर सकते हो. इसे अगर हमको न्यूक्लियोटाइड डेटाबेस में सर्च करना है, ठीक है, तो आप यहाँ पे न्यूक्लियोटाइड को सर्च करोगे. यहाँ पे जो भी आप सेलेक्ट कर रहे हो, आप यू कैन सेव योर रिजल्ट और यू कैन क्रिएट RSS. RSS is similar to that of subscription. Subscribe to RSS for this query, है ना. अगर मैं ये Z92910 का RSS अगर कर देती हूँ तो इससे क्या होगा, RSS is similar to that of सब्सक्रिप्शन. ठीक है, न्यूज फीड भी हम इसको बोलते हैं. तो आपने जो भी क्वेरी का अगर RSS क्रिएट कर दे, इफ यू हैव द लॉगिन ID ऑन EMBL, EBI, देन ओनली यू कैन क्रिएट RSS. तो क्या करेगा, इट विल गिव यू द नोटिफिकेशन. अगर ये पर्टिकुलर 9291 Z92910 में अगर कोई भी मॉडिफिकेशन होता है या फिर इसके ऑथर ने अगर कोई नया सीक्वेंस अगर सबमिट किया है EMBL, EBI में, तो यू विल गेट द notification. Okay, so by creating RSS field, you will notify for any new publication, for any new material or updates on that particular query. आपकी इसी क्वेरी पे अगर कोई भी अपडेट या फिर कोई नया सीक्वेंस सबमिट होता है तो आपको नोटिफिकेशन आएगा. ठीक है, जैसे अब मैंने यहाँ पर Z9, इवन दो यू आर ऑन द न्यूक्लियोटाइड सीक्वेंसेस, सी यू आर फाइंडिंग द क्रॉस रेफरेंसेस हियर, है ना. जीन एक्सप्रेशन है, लिटरेचर, मेटा जीनोम का. आप न्यूक्लियोटाइड डेटाबेस में हो तब भी आप दूसरे डेटाबेस में स्विच कर सकते हो. Okay, you can extract the sequence in EMBL format or in the FASTA format. Okay, this is the European nucleotide ARCHI, which is the nucleotide database of EMBL, EBI. See, this is the sequence version Z92910.1, and this is the description of this sequence. Name of the organism, from where the sequence have been extracted, the accession number, molecular type, topology, base count, data class, division, keyword, chromosome number, then checksum is the software. Then you can view the sequence in EMBL or in the FASTA format. You can download, you can navigate the page, you can see the publication and you can see the sequence version. Navigation में आप click करोगे तो you will find the cross references link. Okay, यहाँ से आपको क्रॉस रेफरेंसेस लिंक मिल जाएगी। जैसे अगर मैं यहाँ पे पीएमसी में अगर क्लिक करूंगी तो मैं कहाँ चले जाऊंगी? आई विल गो टू द लिटरेचरल डेटाबेस। यूरोप पीएमसी इज अ लिटरेचरल डेटाबेस ऑफ EMBL EBI. See, this is the article on which this particular sequence Z92910 has been published. है ना? ये आपको मिल जाएगा. To see how to cite this particular article, you can go to get citation. तो वो पर्टिकुलर आर्टिकल कैसे साइट करना है आप अपने पेपर में आप ये देख सकते हो यहाँ पर. You can also open the PDF of this particular paper. So this is the PDF of the article, full PDF. Okay. Then if you click on the version, it will open up this page. Accession number, accession version, sequence version, first published, last published, view. You can view the sequence either in the EMBL or in the FASTA format. See, EMBL format, word ID stands for identification. It consists of 6 subfields: accession, identification, molecular type, class, division, and base count. The identification name is similar to that of the accession number. Then topology. Then DT stands for the date at which the sequence has been created. Second DT is the date at which it is last updated. D is a description. KW is the keyword used to index the article. OS is the organism source, organism classification. Then RN is the reference number. A sequence entry may contain multiple references, okay, that support the information present in the sequence record. Then DR are the cross references. FT is the feature table. And then last is the sequence, which starts with the word SQ and end with double lash. Okay, this is the EMBL format. If you click on the FASTA format, the FASTA format starts with the sign greater than, then there is a small description, and then there is a sequence. Okay, यहाँ पे एंड में कोई डबल लैश नहीं रहता है. Okay. Then next, if you want to see the publication, तो ये आपका आर्टिकल आ जाएगा, the name of the article in which this particular sequence has been published. Okay. The next is, if you want to submit your data to EMBL EBI, you have to click on submit to ENA. It will open up this page, submitting and updating data to ENA. Okay. So you can submit your data by using the Webin portal or by using the Webin CLI. Okay, Webin portal जो है डायरेक्ट ऑनलाइन होता है, वेब बेस्ड टूल, एंड फॉर द CLI you have to download and use CLI and then submit it to ENA. ENA allow the submission by a 3 route, each of which is appropriate for the different set of submission type. First is the interactive submission, command line submission, and the programmatic submission. These are the 3 routes by which you can submit your data. This table outlines what can be submitted through each submission route, which this we have already discussed in my presentation. Study, sample data, read data, genome assembly data, transcriptome assembly data, template sequence, and other analysis data. Okay. Then to submit your data by using Webin portal, you can go to the Webin portal, and here you have to register first. Okay. यहाँ पे आपको register करना है. You have to make the login ID in Webin, and then you can go for the submission. Okay. Next is support guideline. If you don't know how to use ENA, or if you have any query, you can go to the support guide. Okay, there is a guideline and tutorials which will help you to use ENA. Okay, if you have any query regarding how to submit data to ENA, you can click here. There is various links, है ना, आप अपने query के according link को click कर सकते हो. Then ENA data discovery and retrieval for the data updates. You can click here, and if you are very new and you don't know how to use ENA at all, then you can go to the introductory webinar. This will help you to how to use EMBL ENA. Okay, how to explore on EMBL ENA.